Hello and welcome back, friends. Welcome to Pneumonia. And today we are going to talking about another important topics of molecular biology, that is the DNA is the genetic material. So before going to investigate about that the DNA is the genetic material, first of all we should know the properties of the genetic material. Actually, genetic material actually contain the information of an organisms, and also the genetic material have the property of reproduction, that is they can replicate, and also the genetic material have the property for transmitted from generation to generation, and also the genetic material have the property of the mutation or the changes. So the chemicals which actually contain the following properties, the above mentioned properties, these chemicals will be genetic material. And scientist was confused that whether the protein or the DNA, which one will be genetic material, and due to the diversity of the protein, the various functions of the protein, scientist was confused that the protein may be genetic material. But in 1928, a British scientist, Frederick Griffith, Frederick Griffith actually made an experiment. Frederick Griffith made an experiment. This is bacterial transformation experiment. Bacterial transformation experiment. So what is bacterial transformation? Actually, bacteria can reproduce sexually, and this is a kind of sexual reproduction. The bacterial transformation is a kind of sexual reproduction, and in these cases, a bacteria, suppose this is a bacteria, and this bacteria actually take up the genetic material. This bacteria take up the DNA from outside, so DNA from outside, and this enters into the bacteria and changes these bacterial properties, and so this is called transformation. Okay. And Frederick Griffith was actually experimented on the Streptococcus pneumonia, a kind of the bacteria and which actually causes the pneumonia disease, and now this is called the Streptococcus pneumonia. And this Streptococcus pneumonia he choiced actually the two strain of the Streptococcus pneumonia. So Streptococcus pneumonia have the two strain, Streptococcus pneumonia. So Streptococcus pneumonia, he choiced the two strain, one is the R2 and another is the S3. Okay, and this R2 is the rough variety, and this causes the no disease, so that they are avirulent. Clear? Since they do not causes any disease, so they are avirulent, and this actually causes disease, and they causes disease, and so they are virulent. Okay, and this strain, R2 strain, is actually so this is the R2 strain, so no capsule is actually present here, but the virulent strain have the capsule, so outside there is a polysaccharide coat, and this is the capsule, so this is called the smooth variety, so it has the polysaccharide coat. Okay, so this is the polysaccharide coat, and this polysaccharide coat, due to the presence of this polysaccharide coat, this is called the smooth variety. Here I shall tell you that a key point, that is R2 can be mutated into S2, never S3, never S3. So the mutation is strain specific. So mutation is strain specific. Okay, and now let us examine which experiment. Now let us go into the examination of Frederick Griffith. Actually, show that the Frederick Griffith actually made the experiment of bacterial transformation, and now I am going to explain you about the bacterial transformation experiment. So first of all, he actually take up the R2 strain, and when this R2 strain inject into the mice, so inject into mice, the mice will survive. Okay, the mice will survive, and when S3 strain inject into mice, the mice will die due to the disease. And when heat killed S3, actually inject into the mice will survive. But when heat killed S3 and plus R2, when inject into the mice will die. Frederick Griffith was surprised that whether, and here you can see that heat killed S3 actually causes the survive, but heat killed S3 and the R2 and both actually do not causes the any disease. But when the both these strain actually injected into the mice will die by the disease. So what will be the reason behind it? Actually, the reason may be that since the R2 cannot be mutated into the S2 because the mutation is the strain specific, so there is no mutation. So no mutation. Transform is R2 into S3. R2 into S3. Then why S3 found? Then why S3 found? And this is a question. Okay, and Frederick Griffith actually guessed that a chemical, and he gave his name as the transforming principle, and unknown substance, transforming principle from S2 from R2 transform into the S3. So a transforming principle actually transformed R2 into the S3, and he could not find out this transforming principle. And Frederick Griffith actually performed this experiment in 1928. But later, three scientists, Avery, MacLeod and the McCarthy, was trying to investigate about that which one is the transforming principle. They was trying to find out that which was transforming principle. So, and this is the bacterial transformation experiment performed by the Frederick Griffith. And now we are going to explain you about the conformation experiment. Which one is the genetic material? Frederick Griffith actually supposed that the protein may be the transforming principle, but in 1940, Avery, MacLeod and the McCarthy made an experiment. So in 1940, Avery, MacLeod and McCarthy. Avery, MacLeod and McCarthy made conformation experiment. Okay, so conformation experiment. Clear? And in these cases, which one was that, and what was their experimental procedure? So that they actually take a test tube, and in this test tube they take R2 and heat killed S3, R2 and heat killed S3, and they take up many test tube and show that the R2 and the heat killed S3, and when in this test tube, so if this is the R2 and if this is R2 and this is the heat killed S3, suppose this is heat killed S3, and they actually gave in a test tube, suppose this is the test tube. I have a drawing is as that I have drawn it as the normal single test tube, and if we the protease in a test tube, then the S3 colony can be found, and if we give the amylase, S3 colony will be found, and if we give the RNase, then the S3 colony will be found. But if we give the DNAase, the S3, no S3 colony. Clear? So what is the what is the conclusion for this experiment? Actually, the DNA of H3, the DNA of H3 enters into R2 and transform it into H3. Okay, so the transforming principle is the DNA. So DNA is the genetic material, and in the first cases when we added proteins, then the protein will be break, but the DNA remains the intact, so that the H3 colony can be found. But when we give the amylase, carbohydrate will be break, but DNA remains intact, so that H3 colony can be found. In the RNAs can be break, and DNA remains intact. But when we add the DNAs, then the DNA remains breakdown, so DNA will be damaged, so that the DNA of the H3 bacteria cannot transform the R2 bacteria into the H3 colony. So the DNA is the genetic material because it has the property of changes a bacteria into that of the another phenotypic character of the another bacteria. So this is the transforming principle, and the DNA is the transforming principle, so that the DNA is the genetic material. And another experiment was performed by Hershey and the Chase. They made an experiment of the transduction. So what is the transduction? When the DNA from any of that of the bacteria is actually enters into the another bacteria through the bacteriophage, and this process is called as that of the transformation. Suppose this is a bacteria. Suppose this is a bacteria, and bacteria 1, and this is another bacteria. So another bacteria, and if the DNA from this bacteria is actually enters via the bacteriophage, suppose this is the bacteriophage, so this type of reproduction is actually called as the transduction. So this is called as that of the transduction. So this is called as that of the transduction. And here in the transduction, a bacteriophage. So this is the bacteriophage. A bacteriophage actually carry the DNA from one bacteria to the another bacteria, and this transform this bacteria, and so this is actually called the transduction. And the Hershey and Chase perform another experiment which actually clearly proved that the DNA is the genetic material. Now let us study about the Hershey and Chase experiment. So that Hershey and Chase bacterial transduction experiment. Hershey and Chase experiment. Hershey and Chase experiment. Okay, and transduction experiment, and this is transduction experiment. Clear? And in the bacteriophage, suppose this is the bacteriophage, and this bacteriophage have the protein coat, and they actually added in this protein coat in the 35S. So this is the radioactive element, radio-labeled, and in this protein, so protein was labeled with the 35S, and here is the DNA. Suppose this is the DNA, and this DNA was labeled with 32P. So DNA labeled by P32. Clear? And they actually chose the T2 phage, and which infect E. coli, clear? And when this actually infect an E. coli bacteria, so what will be occurred? So this is the E. coli bacteria. Suppose this is E. coli strain bacteria, and this bacteria is infected into this, and so 35S, and the red one is the 35S, and here is this is the 32P, and here is the 35S. So 35S, and they actually, and by the process of the of the lysis, and this DNA actually enters into this bacteria, so that the DNA enters into the bacteria, DNA labeled with the P32, and when these bacteriophage actually lies these actually lyses from this E. coli cell, or come out from the E. coli cell, the no 35S so no 35S can be found, but the radio labeled 32P, which was labeled by the DNA, so 32P DNA can be recovered in the phage progeny. Okay, progeny. So DNA is transmitted from generation to generation, and this is an important property of genetic material, so that when we labeled the DNA with the help of the P32, because the DNA has the phosphorus, and when the protein is actually labeled by 35S, because the proteins have that of the sulfur containing molecule, so that and when this actually infected into the E. coli bacteria, and then the progeny, by the process of that of the lytic cycle, and the progeny which are actually formed, and they actually contain the 32P, and which was present in that of their parent phages, so that this 32P or the DNA is actually transmitted from these parents to their progeny. So the DNA is the genetic material. So that I have described to you about the bacterial transformation principle, which was performed by bacterial transformation experiment, and which was performed by Frederick Griffith, and next the conformation experiment performed by Avery MacLeod and the McCarthy, and whether the Frederick Griffith could not identify the transforming principle, or which chemicals actually transform a bacteria to the another bacteria, that is R2 bacteria to the another strain of that of the Streptococcus pneumonia into the S3, and he could not find out the chemicals, and he actually gave its name as that of the transforming principle, and he was supposed that it may be protein, but Avery MacLeod and the McCarthy actually confirmedly proved that this transforming principle is the DNA, and later Hershey and the Chase actually experiment, and this actually clearly proved that the DNA is the genetic material. Most of the organisms, actually from the prokaryotes to the eukaryotes, every organisms, the DNA is the genetic material. Even in cases of the viruses, the DNA containing viruses, DNA is the genetic material, but the few viruses only in this world, only the few viruses which contain the RNA, and the RNA is the genetic material in this kind of the viruses. So that just like the tobacco mosaic viruses, the RNA is the genetic material, and apart from that of the RNA containing viruses, every organisms, the DNA in every organism, DNA is the genetic material. Okay, and this genetic material actually they carry the information or the blueprint of an organism, and this actually transform from generation to generation, and this has the property for the replication and also the properties of the mutation. So that's it guys, and if you like my videos, then you can subscribe my channels for more updates. In the next video I shall describe you about the structure of the DNA. So thank you guys.